Welcome all. So I am Dr. M. Thiruselvi from Dr. N.G.P Arts and Science College. So today we'll have a brief discussion on monoclonal antibodies. We'll be discussing about what is mainly monoclonal antibodies, what is hybridoma technology, and what is the methodology behind hybridoma technology, and what are the steps involved in it, and what are the applications of monoclonal antibodies. So before we go in for what is meant by monoclonal antibodies and its production, we need to know what is meant by antibody. So antibodies are also known as immunoglobulin, which is a protein molecule which is used by the immune system to identify and neutralize the foreign substances like virus, bacteria, fungus, etc. So monoclonal antibodies are identical immunoglobulin which is generated from a single B cell clone, and these antibodies will recognize a unique epitope or a binding site on a single antigen. And there is another term called polyclonal antibody, and these polyclonal antibodies are mixture of heterogeneous which is usually produced by the different B cell clones in the body, and they will be able to recognize and bind to many different types of epitope of a single antigen. So monoclonal antibodies are homogeneous clone of antibodies, and they rise from a single plasma cell and they bind to a single epitope of the antigen. And these monoclonal antibodies are produced in a specialized cell through a technique known as hybridoma technology. And first, the production of monoclonal antibodies was first introduced in the year 1975 by Cesar Milstein and Georges J.F. Kohler. Next, moving on to what is meant by hybridoma technology. So the term hybridoma technology was coined by Leonard Herzenberg in the year 1976 to 1977. So what is meant by hybridoma? That is a fusion of the plasma B cell with the cancerous myeloma cell to produce a hybrid cell known as hybridoma cell. And the production of hybridoma cells is known as hybridoma technology. They are immortal and they live for a long time. Next, moving on to the production of monoclonal antibodies and what are the steps involved in it. First, the immunization of an animal. The mice being exposed to a particular antigen for which the monoclonal antibodies are to be produced by intravenous administration. So, respective antibodies are synthesized against that particular antigen, and spleen is being dissected and splenocytes are isolated from the mice and plasma B cells are cultured. Next, after the immunization, cells are being fused. The activated plasma B cells are fused with immortalized myeloma cells in the presence of polyethylene glycol solution. So, as a result of this cell fusion, they produce the fused plasma cell, unfused plasma cell, fused myeloma cell, infused myeloma cell, and the hybridoma cells. So here is the diagram which tells you about how the mice is being immunized with a particular antigen, and as a result of this the antibodies are being produced, and once the confirmation of the production of antibodies, the spleen is being isolated and the spleen cells are being isolated and it is being fused with the myeloma cells, and these fused hybridoma is being cultured in a hack medium and it is selected for the positive cells. Finally, the monoclonal antibodies are harvested. After the fusion, the hybridomas are selected in a hat medium. So the hybridoma cells are selected by incubating the fused cell in the hat, hat medium, which is hypoxanthine amnopterin thymidine medium. Due to the presence of amnopterin in the myeloma cells, that blocks the very important enzyme, that is dihydrofolate reductase, which is important in de novo pathway for the synthesis of nucleotide. Cells which lack HGPRT, that is hypoxanthine guanine phosphoribosyl transferase, do not grow on the hat medium. So HGPRT is a precursor for the nucleotide synthesis in salvage pathway. So salvage pathway is the alternate pathway for nucleotide synthesis. So only the hybrid cells can survive because they possess both the property of plasma cell as well as the myeloma cell. Next is the isolation of hybridomas of single specificity. The incubated medium is then diluted into multiple plates to such an extent that each cell will contain at least one cell. Next is the screening of the product. Then the supernatant in each well can be checked for the desired antibodies, that is the monoclonal antibodies, by two types of screening, namely ELISA and RNA. After screening of the product, next is the cloning and propagation. So now we have a separate clone, that is the activated B cell, which produces the antibody of a single specificity. Hence the number of monoclonal antibodies are harvested. And next, after the cloning and propagation, it has to be stored. So it is stored in a liquid nitrogen. Under in vitro condition, the incubated medium is then diluted into multiple plates to such an extent that each cell contains at least one cell. Then the supernatant in each cell can be checked for desired antibody, that is a specific monoclonal antibodies. Hence, more number of monoclonal antibodies are harvested. So, next, what are the advantages of monoclonal antibodies? So, it is highly specific recognition for only one epitope of an antigen, and it is immortal hybridoma cell lines have the ability to produce unlimited quantities of antibodies, high consistency among experiments, minimal background noise and cross reactivity, excellent for affinity purification. Next, move on to the applications of monoclonal antibodies. It tracks the cancer antigen, so it is very important in the cancerous studies, and it also helps to distinguish between the B lymphocyte and T lymphocyte, and it also used to locate the cancerous tissue. And these monoclonal antibodies are used to diagnose the disease like gonorrhea, and it is also used in the fluorescent immunoassay, and it is also an essential tool for biomedical research. So in this video, we discussed about what is meant by monoclonal antibodies, and what is meant by, apart from this, what is meant by monoclonal antibody, what is meant by antibodies, etc. And what are the, how this monoclonal antibodies are being produced using hybridoma technology, and what are the steps that are being involved in hybridoma technology. That is, first is the how you immunize the mice with that particular antigen. Then after you see for the production of antibodies, then you isolate, dissect the spleen from that particular mice, isolate the spleen cell, and fuse the plasma B cell along with the myeloma cells and allow it to grow in a hat medium. Then then finally harvest the monoclonal antibodies, and what are the advantages of monoclonal antibodies, and what are the application of monoclonal antibodies in research as well as in the medical field. Thank you.